I'm going to try to get you started on your X5 genetic disorder analysis project as quickly as possible. So if you try to follow me, I'm going to open up 5 pages, get their links, and then walk you through how to read those pages. Specifically, I'm going to imagine that I'm doing a project based on a gene called PRLR, the prolactin receptor. So first thing I want to do is open up all 5 of these pages, which are linked on your information document for this project. But again, the top 5 that I would start with are the NCBI gene page, UniProt, ClinVar, OMIM, and the PDB protein databank. So what I'm going to do is I'm just going to type in PRLR into each of these and get my results. And I'll do this as quickly as possible. And then I'm going to take all of those links and paste them into my sort of starting document so I have them available to me. Oh, and it looks like I already did on this one. So here we go. So NCBI, here's the gene page. UniProt, let me go to the human one, and there it is. Here's ClinVar. Here is OMIM, all set, and structure, here we go. So what I'm going to do is I'm going to grab each of these pages, grab the URL, paste them in here. I could just paste the URL. I'm actually linking this so I don't get lost or forget what's what. So that was this one. Here's UniProt. Copy and link. And again, the thing is, like, you're going to be doing this project over multiple days. And so what you don't want to do is have to find these pages over and over and over again. So I am unfortunately wasting a couple minutes of your time so you can watch me actually copy links down. All right, almost done. I'm just going to stop here and move on. So get those links. It'll help. All right. So we're starting with the gene called PRLR, which you might not know anything about. I know almost nothing about. But let's start by trying to collect as much information as we can as quickly as possible. I'm going to try to get the chromosome number, the number of exons and introns, where it's expressed, how many isoforms, if any, there are beyond one, the parts of the protein known as the domains, and we'll pick an allele associated with some sort of disorder. So let's start here. Okay, we're going to start with the gene page. Here is the human PRLR prolactin receptor gene page. You'll want to read this. I'm not going to spend time looking at this right now, but this is important. The summaries where they explain what the protein does is kind of the basis for this whole project. But for right now, let's just collect information. On the right, you'll notice there's a table of contents, which gives you quick links to different parts of this page because this page can be overwhelming. However, I can also close any one of these sections pretty quickly. If we start at genomic context, I can see which chromosome it's on, and there it is. PRLR is on this particular strand of chromosome 5 facing in this direction, and so I might as well write that down. It is on chromosome 5. Done. Now, I will have to cite this. I'm not going to walk you through that. That will have to be a separate video, but that's our first step. I can also see where it is on the chromosome. If you really care, it happens to be at nucleotide, approximately. Nucleotide number 35,048,756 is where it begins and goes for quite a while. 200,000 base pairs almost. Pretty long gene if you include all the introns. All right, next thing. I got 16 exons. It says right here. This isn't always right. This is automated in terms of how it's collected. But for right now, let's just collect this information. We can analyze and cross-check to make sure that's correct later. All right, if there are 16 exons and there's an intron between the exons, that means there would likely be 15 introns. There are exceptions, but let's start with that. Okay, where is this gene expressed? Meaning, where's the protein produced? Where's the prolactin receptor produced? I'm going to skip down to a location here called the expression location, and it actually will, through a number of different research groups here and research studies, suggest where this is shown in your body. And it's a listing right here: adrenal gland, endometrium, kidney, and most importantly, the placenta. Now, I don't know if this is going to necessarily match with what you're expecting. Honestly, I'm expecting the prolactin receptor to be most highly expressed in mammary glands, breast tissue. But that wasn't actually listed here. Okay, it's not one of the things that was studied in this particular research project. But for the purposes of this project, I might as well list, well, the places where it was most expressed according to this particular page is placenta, followed by kidney, adrenal gland, and the endometrium, where placenta was the highest. But that's just one link. Another spot that you'll probably want to go to, which I forgot to open earlier, is the Human Protein Atlas, and let me open that right now. And this is probably a better source of information for where your particular gene is expressed. On the Human Protein Atlas, it will give you a number of different things. As it loads later, I will then go to it. But let's keep on moving. Okay, how many isoforms are there? When you express this gene, is there just one version of an mRNA that's formed or multiple? So I'm going to close a bunch of these tabs, a bunch of these sections that I don't need to look at, and I'm going to go all the way down to the reference sequences. Again, there is a link on the right. All right, the reference sequences are both the chromosomal information, which you don't want. You don't want all the introns and stuff. And here we go, mRNA and protein sequences. Here's one. This is one version of an mRNA followed by the polypeptides that's generated from that mRNA. If I scroll, I can see that there are multiple different mRNAs that have been found and sequenced and put into this database. In fact, there appear to be 6 different mRNAs that have been found in people. So I might want to note as a starting point that there might be as many as 6 different isoforms. Now, are any of them important? I guess that's the next thing to look at, which is to see if they actually differ in any way. And so right here, I can see this variant is the longest. Okay. Next one, isoform 6, which is number 2 on the list. These might be ranked in order of how common they are. That's my assumption, but I don't know. All right, next one. This variant lacks 2 in-frame coding exons. Okay. Lacks 2 coding exons. That's a pretty major difference. That would suggest that this polypeptide here is very different from this one. So not only are they multiple isoforms, but it appears that they might and probably do encode at least 2, possibly more different polypeptides. So that will be something to look at later. Again, I'm just getting my start on this project. Next, let's find the domains of this protein. Best place to go for this, let's go to UniProt. So this is the UniProt page, and there is an immense amount of information on these pages. Again, there's a table of contents. In this page, it's on the left. All right, scrolling down, we can find the function summarized here, what it's good for. This summary isn't especially great, but I'm just going to move on. It's going to tell us what certain positions, meaning amino acid residues, places in the protein, what they're for. This apparently binds a zinc ion, so it's got a metal ion cofactor that helps with the structure. Okay, let's move on. I'm going to skip a bunch of this, but I'm going to try to get parts of the protein and their functions. Now, right here, this is kind of interesting. Subcellular location, where do you find the protein? Well, it says that it's secreted. So it's not in the nucleus, ER, mitochondria, Golgi, any of the internal organelles. It's taken, probably produced in the ER, vesicle, Golgi, vesicle, and sent out. All right, maybe it's a membrane protein. In fact, it says membrane protein. So this is a little bit confusing. It's saying secreted, which suggests it's being sent out of the cell completely, but it appears that this is a membrane protein. So it's going to go to the cell membrane out here. Let's keep reading, see what we can find. But it might be noting that this is probably a membrane protein. Okay, scrolling down. And now we have confirmation. So right here, we've got some information that the first 200 amino acids of the protein, other than the first 25, are outside the cell. The next stretch of 24 or so are in the membrane, going through the membrane. They are our anchor into the membrane. And then this massive region on the inside, on the cytosolic side, has another function. And this is going to be a typical receptor shape, right? You've got your, for example, a sort of a binding pocket on the outside, your catcher's mitt for the hormone, then something that's attaching it to the membrane, keeping it in the membrane, and then this cytoplasmic region is going to be what starts the signal transduction pathway. Now, this doesn't appear to be a G protein coupled receptor, which would go through the membrane typically 7 times. This doesn't appear to be a RTK because they would probably say that this is a kinase region on the inside, so it's a different type of receptor that you might not have learned about before. My guess is this is a cytokine receptor. Again, nothing you need to worry about right now. You'll learn about it as you progress through this project. Okay, so we got 3 domains already. So at minimum, we have an extracellular domain, again, your catcher's mitt. We've got the transmembrane, and then we have the internal domain, which is going to start the signal transduction process. That's what receptors do typically. So I'll list these. So under domains, we've got 3 at least. So we've got at start, and let me just bullet point these. Get this over here, 25 to 234. Can I copy? I can. That'll be our extracellular domain. And that's probably our receptor location. Then we have the part that's going through the membrane right here. And you can see what I'm doing here. I'm not going to collect all of this information in real time for you. That's like not going to be a good use of time. Hold on. So this is going to be the transmembrane domain. And then I'll just list this next part really quickly. I'm not even going to get the numbers right. Well, maybe I'll do this correctly. All right, 259 to 622. And this is going to be the cytosolic portion or cytoplasmic location. So we got 3 shapes. We're expecting a big shape outside, a thin shape going through the membrane, and then a massive shape on the inside. Hopefully this is getting you a good start as to like how this protein works. Your protein's going to be different. We're all going to see what our proteins do separately. I'm scrolling down. I'm not looking at every single section here. I'm just scrolling down to see what other information I can get. Hey, 1 to 24. That's a signal peptide. Great, let me note that. If you don't remember, what a signal peptide is the portion of a new polypeptide coming out of the ribosome, a ribosome that is, that targets it to the ER so that the protein actually gets either translated directly into the ER lumen or into the membrane, or a little of both. And so this is going to be why the protein ends up at the cell membrane, is because of the signal peptide. So that explains what the first 24 were. So now we have the basic domain structure of this whole thing. The beginning's the signal peptide, then you've got your binding pocket, then you've got your transmembrane, then you've got your portion that starts the signal transduction pathway. We do have some other details here. Apparently at 3 different sites here, 59, 104, and 233, there are some carbohydrates added. We haven't talked about this a lot. Glycosylation is the fact that you can add certain branched carbohydrates to the outsides of some proteins. This is really common for membrane proteins, incredibly common for membrane proteins. So that's on the extracellular receptor side. And so I would maybe note multiple glycosylation sites. And in reality, I would probably have listed the actual numbers here. But I'm not going to bother wasting your time as I do this in real time. All right, that gets me the shapes, all right, the different portions of the protein that are doing things. Next, let me see what I've got next. We got to pick an allele. So we got to pick one version of this gene that's causing problems. And so our 2 sites for that are going to be OMIM, here's one, and the other one's going to be ClinVar, which is right here. Now ClinVar, this is an NCBI database. What it does is it actually has data from individual human beings. And so if I scroll, every one of these listings is an actual human being whose gene sequence for this, and maybe their entire genome, has been sequenced. And so it's been posted here. What I want to do, though, is just look at the start up here. From left to right is the length of the gene, and all of these little dots in color represent locations where there's variation known in humans. And so each of these dots, again, is a location where there tends to be some variation. Some of these don't seem to make any problems. So anything in dark green, light green, are not known to cause any particular symptoms. Those things in red are known to be associated with particular symptoms or disorders. So I want to ignore all these other ones and focus specifically on the ones that are problematic or pathogenic in this case. So I'm just clicking the filter, which I actually had clicked before I unclicked it for a second. All right, so here we go. All of the problematic ones appear to be in this one location of the gene, which appears to be towards the beginning. And if you remember the beginning of the gene, after the signal peptide, this is the extracellular portion of the protein. This is what encodes the extracellular, the receptor binding pocket. All right, if I scroll down, we can see the different versions of this gene and what has happened to it. Now, I'm going to ignore all these that are copy number loss or gain. That means that the entire gene is missing or has been duplicated. That usually means some crazy crossing over error, and this is going to have wildly varied and multifactorial impacts because it's not just one gene that's being messed with. I'm skipping all these. Ignore those, please. We want to look at things that affect, under gene, just the gene that you're interested in. I've only got 3 possibilities here in this database. I've got one here, which is a single nucleotide variant, which is a C to a T, as you can see here, which changes a single amino acid from a proline to a leucine. This one switches a histidine to an arginine. This one switches an arginine to ter. That means terminal or terminus. That's a stop codon. So any of these seems like a reasonable thing to study. I could find one that messes with one particular amino acid. That seems like a perfectly reasonable thing. All 3 of these appear linked to this disorder called hyperprolactinemia. Great. So I'm going to choose one of these. Ideally, what I would do is go to this OMIM page and find one of these that has a good amount of information in. Specifically, towards the bottom under alleles, they tend to have a good amount of information about the families that might carry these alleles and some of the symptoms that result and what they know about how this disease is or disorder is passed on. Up at the top, it even shows whether it might be autosomal dominant, that's AD, or autosomal recessive, or depending on the allele, maybe one or the other. So this one's a little complex, it appears. But we'd have to do more research into that. So I've got multiple alleles listed here, 4 that have been selected for this page by the editors, and 1, 2, 3 that are associated with this disorder, hyperprolactinemia. Okay, so if I had more time, I would read this. But for today, I'm just choosing one of these 3. Let's just choose this one right here. 635 A to G. So 635 goes from an A to a G. And the polypeptide switch is a histidine to an arginine at 212. All right. Histidine to arginine at 212. R stands for arginine, but I guess I can write that out to make it more obvious. Okay, so it goes there. There we go. And you can see the original and the mutated version. Okay. The disorder that's associated with these, hyperprolactinemia. And we can study more of that in a bit. Again, we're just listing as much stuff in 25 minutes as we can to get started on this project. Let me get some alignments, then I'll pull a structure. When I say alignments, I mean an alignment of sequences to see how, I want to see that A to G change. I want to see where the histidine to arginine is. So for this, I'm going to go back to the original NCBI gene page. Here it is. I'm going to open up the mRNA page. and the protein page, the polypeptide page. And so I'm opening up the most common isoform, which in this case is 1. In your case, hopefully that's correct. That'll make this the easiest possible thing. And so at the bottom of each page, you can actually see the sequence, letter by letter. All right, this is the DNA version of the mRNA. So you can just think of this as the mRNA sequence, just with T's instead of U's. It's long, and we don't want to look at this whole thing. There's no way. What I would advise you to do, first, from the top, scroll down past all the references. You might want to read the summary, though that's probably similar to what you saw on the gene page. But I'm going to scroll down. There's exons listed. CDS is what you need. CDS stands for coding sequence. Click on that. This is the actual recipe. From the ATG start codon all the way down here, there's your stop codon with the TGA. I could copy and paste this whole thing, right? In fact, there's no harm in doing so. Maybe I'll just do that to show you that. But you're probably not going to want to include this entire thing in your paper. But again, if you're just collecting information, sure, let's grab it. Next, what you might want to do at this point is thinking, well, where's this 635A changed to a G? So we can find that. We really can. So let me get this a little bit more compact if possible. There we go. And so I'm looking for 635, you'd think. So here we go. Here's 601 right there. And then these are in blocks of 10. So 611, 621, 31, 1, 2, 3, 4, 5. And that is actually not the A we're looking for. It was a 1 in 4 shot. It just happened to be an A. Here's where it gets a little complex. This A right here of the ATG, this A of the ATG is the first nucleotide in the coding sequence. That should be number 1. When it's listed as a 635 mutation, that's where this is 1. So try to follow me on this. Feel free to rewatch as needed. If I want to figure out where that 635 A to G is in here, I have to renumber. So in this particular mRNA, let's see, number 1, the nucleotide that should be number 1 of the coding sequence is listed here as 301, 311, 321, 22, 23, 24, 25, 26. So it's actually 326 that on this numbering system, that is number 1. And I'm looking for 635. So to find where 635 is in here, I'm going to need to add 325 to it. If I do that, I should find the mutation location. So this should be at 9. Oh, it's doing the math for me. Thanks, Google. So A635 is actually at 960. So here's 961. Let me go back 1. 960 is right there. So let me bold that. Let me undo this, or let me hide some of these letters. I'll just make them very light. I'm going to change all of this into a reasonably readable something or other. I'm going to get rid of the highlight, get rid of these letters which are past the stop codon. And now we've got the gene sequence with the mutated A, which becomes a G in this particular family, posted. Now, if I want to actually make this a little easier to read, what I might do is the following. And I might suggest you do this. I might delete all that, copy, paste, G. That's the mutation. All right, so this, what I've just created, is a partial sequence of the mutant allele, or the allele itself, that is associated with hyperprolactinemia. Let me do the same for the polypeptide sequence as quickly as I can. Oh, closed the wrong thing. Oh well. So here's the protein, or the polypeptide sequence. Copy that. Paste it. This is actually numbered correctly. There's no start codon thing to worry about. So histidine 212 is going to switch to an arginine. So 212 is going to be, so here we go, 181, 191, 201, 211, and that means there's the histidine. So if we want, and I can make that a little more obvious, or I can even highlight it, I could, if I wanted to show you what the mutant protein would have, I could do it this way, histidine to arginine, and now I have the full length wild type isoform, or most common isoform right here, and here's the mutant allele right here. Or if I want to make this easier to read, because it's only one amino acid different, the rest of the protein isn't changed, it's not a stop codon or something. So I might just do this and show there's just a single amino acid different between these two alleles. I probably want to note that this is the wild type, and this is the mutant allele that we're talking about. So just line them up like so. Same thing up here. And keep in mind, there's not like just one mutation. Like this is just one particular allele that we're looking at. This isn't one of those capital B, little b things where there's only two varieties. There's lots. But hopefully this gets you a start on laying out your alignments. Okay, lastly, as quickly as I can, because this video is already longer than I was hoping, structure. So, two places that you might want to go. The easiest to deal with is UniProt, because it will put multiple structural images together for you in this plugin right here, and it's loading the first. In UniProt, it will actually have all of the structures that are known for this protein alone, listed here. Now here's where to be careful. Where I'm pointing to, you can see which amino acids are in the structure. So when they did X-ray crystallography for this, they did not attempt to make a crystal of the entire protein. Doing that with membrane proteins is very difficult for reasons that I don't have time to get into. But the protein that they have is 25 to 235. And so if we look back here, they have the extracellular region. So they've got the binding pocket for grabbing that prolactin hormone. Great. That's something. And that might be helpful enough because the mutation we're looking at is before 200. It's 212. So it would actually be in this region of the protein. Now, ideally, we'd have the whole protein. But if we scroll these, nearly every one of these is only the extracellular portion. We can look elsewhere. We could go to the protein data bank here, and I could type in PRLR and get my results here. And there are multiple hits. And maybe they have, here's the transmembrane domain itself by itself, just that portion that goes through the membrane. We do have human prolactin stuff, it appears here. So maybe I could find some other stuff. But for right now, let's keep this simple. I'm going to choose one of these structures here that covers the extracellular domain. Notice most of these don't have the 1 to 24 because that's snipped off when you have the mature polypeptide. Any of these is just fine. They all have pretty good resolution. The smaller the number, the better. So sure, let me grab one of the 2 angstrom resolution structures, and I'll just open it as a separate tab. And this is going to be the PDB page. I would definitely want to grab the citation for this, right? I'm going to grab that citation as soon as I get to this page before I go any further. Here's the structure. And this is going to have some sort of antagonist, but we don't have time to talk about that right now. But this protein structure, let's let it pull up, will have the entire extracellular domain of the receptor. So here we go. Oh, there's prolactin. Oh, this is pretty nice. I'm enjoying this. So here's the prolactin receptor. The prolactin receptor has 1 polypeptide in this particular structure, and it's the orange one. We can see it highlighting individual amino acids. And then prolactin is the green alpha helical polypeptide there. All right, so we've got the receptor, we've got the polypeptide, that's prolactin. There might be more to learn about this. This appears to be some sort of mutation in the prolactin or in the receptor. We'd have to look more specifically into that. Now, in this case, what we'd probably want to do is find the specific amino acid that is mutated in our allele. So if we're looking for the H, the histidine at 212, that would be important to find. So if we go back to the structure, where am I? Here we go. We're looking for the histidine at, I've already forgotten, 212. H212. Now, it does not appear to be in this structure. So we got a problem. So we might have to pull another one. So these things happen, right? So we're going to have some troubles in some cases trying to find the specific location. Your job, though, will be to find your alleles like variant location in here as best as possible. I'm just going to pick a different amino acid to highlight so you can see how to use this particular plugin. Things to notice. If you're zooming in and you lose your way, feel free to click here and it'll reset you. If you want to select specific things, I suggest clicking on the arrow, and this right here is going to allow you to choose multiple amino acids to highlight. So if there's an entire region you want to highlight and say, this whole region is destroyed, or this whole region in the mutant version is after a stop codon, use it. All right, this may be helpful to you to highlight specific locations. So if I needed to, for example, highlight just one amino acid and say there's just one arginine or one histidine that's messed up, let's imagine it's this histidine right here. I can click on it. It will highlight. You might see it right there. The other thing I will quickly show you is there are other ways of representing this polypeptide. Oh, that's not the histidine. That's in the prolactin. Hold on. Let me switch to the receptor. I was in the wrong polypeptide. So here we go. So I'm going to click on this histidine right here, and hopefully it will highlight for us. Let me unhighlight the prolactin amino acid, go to the receptor, and I'm just going to pick a histidine right there. Okay. And you may not have an easy time seeing it, but it's right over there. I'm going to help you see these things a little bit better. Now, again, you can click on that. You can zoom far in and see the actual bonds. We did that earlier in the year. I'm going to add something here that might help as well. Under polymer on the right, add representation, surface. I think this is helpful because you can see in a three-dimensional format where that amino acid might be. Now, it's actually tucked away. Right? It is hard to see because it appears to be one of the amino acids that's involved in actually binding to the prolactin hormone, which indicates why this might be a problem if that switches from a histidine to an arginine. And you definitely want to figure out the amino acid difference, like what are the properties between those, because they do differ. So that's the way to highlight. Hopefully that helps you. Make sure that you are able to pull an image from here. You can screenshot from here. I believe that's this. You can screenshot and export images from this, or just do a screen capture yourself using whatever your operating system allows you to do. That will go into here, and hopefully that'll allow you to really start figuring out why the allele that you're studying doesn't work in the same way that the wild type, or I guess normal is the word that a lot of folks would use, what the wild type, most common allele does. See what the differences are. Okay, folks, that was a whole lot. I hope that was helpful. Again, that walks you through how to find the gene, how to see the sequences, figure out where the gene is expressed, how many exons and introns are in there, how many isoforms, and if they actually differ, what are the domains of the protein, picking an allele, picking a reasonable allele, finding a structure, and then how to analyze it. Hope that helped, folks. I'll see you next time.